Welcome to the fertilization module. In this talk, we're going to learn about the structure of gametes. We will first learn about the different gametes that are involved in the process of fertilization, followed by different structural aspects of the sperm and the egg. Fertilization is the process when a male and female gamete fuse to form a diploid zygote. The male gamete is referred to as the sperm, while the female gamete can be called as the egg or ovum. There are many structural differences between the male and female gametes that we're going to learn about, but when we look at their size, they're not equivalent. The male gametes are a lot smaller than the female gametes. Let us now look at the structures of the male and female gametes, and we will begin with the sperm. Now, each sperm has a haploid nucleus, a propulsion system to move the nucleus, and a sac of enzymes to aid the nucleus to enter the egg. Let us now look at the different parts of the sperm. Now, the sperm can be divided into the sperm head, the midpiece, the tail, and the end piece. Now, the sperm head is made of the nucleus and the acrosome. The nucleus is very tightly compressed and haploid. It is shown in the form of the blue circle that is present in the sperm head that's shown in the figure below in the blue box. The acrosome is also called as the acrosomal vesicle, which is a Golgi-derived structure and contains enzymes that can digest the outer covering of the egg. It is shown in the moon-shaped green structure in the figure in the sperm head portion. The flagellum is the propulsion system of the sperm, and that is what is shown in the blue box in the figure. There are different parts of the flagellum, and the first part is the axoneme, which is a motor made from microtubules emanating from a centriole. The centriole is that small yellow dot that is right after the nucleus in the figure that is shown below. Another motor protein called dynein uses ATP and provides the force for propulsion. The ATP that is needed for propulsion is supplied by mitochondria that are shown in the form of those tiny red dots that are in the midpiece of the sperm. Thus, the flagellum allows the sperm to move around so that it can find the egg. The egg or ovum has the material that is necessary for the growth and development of the embryo. When we look at the egg and compare it with the sperm, it has much more cytoplasm than the sperm. The different types of other molecules that are present in the egg are the nutritive proteins that will supply energy and amino acids to the early embryo, ribosomes and transfer RNA that enable the early embryo to make many of its own structural proteins and enzymes, various maternal messenger RNAs that encode for proteins during the early stages of development. Morphogenetic factors are present, like transcription factors and paracrine factors, that direct the differentiation of cells into certain cell types during early embryonic development. And the cytoplasm can also have protective chemicals that protect the egg from threats. The haploid female pronucleus is called as the ovum or the egg. Now, when we use the term oocyte, we normally refer to the female gamete that is still undergoing development and is normally not ready for fertilization. Now, in the case of mammals, the final stages of egg meiosis takes place after the sperm's nuclear material is inside the egg cytoplasm. So, in the case of mammals, the female gamete is not a truly haploid when the sperm's nuclear material enters the egg. When we look at the figure below, we're looking at the different stages of development of the oocyte, and we're also seeing the different organisms listed below each stage of development, which indicates that stage during the process where fertilization occurs or where the sperm enters the egg. For example, in the first one, we are seeing a primary oocyte. The primary oocyte is still undergoing development and has not yet formed the haploid gamete. However, in the case of organisms like dogs and foxes and certain roundworms or certain sponges, this is the stage where the sperm will actually enter the egg. Now, in the case when the primary oocyte will proceed to the first metaphase stage, so we're looking at the first stage of meiosis or meiosis 1. As we saw in the previous chapter, in the case of the developing oocyte, meiosis happens in such a way that the mitotic spindle or the meiotic spindle is actually present in a corner of the cell. Now, in organisms where the oocyte is in this specific phase, that is when the sperm will enter the egg. And so this is seen in certain mollusks, many insects, some starfish, and other kinds of organisms. The completion of the first meiotic cell division gives rise to the secondary oocyte, which is the larger cell. So, the first meiosis undergoes cell division in such a way that the two daughter cells have unequal cytoplasm. One daughter cell has a lot or majority of the cytoplasm, and that is the secondary oocyte. Now, in certain organisms like amphibians, and in the case of mammals, the stage in which the second meiotic division has begun but has not yet been completed is normally the stage at which the sperm will enter the egg. When we look at the final stage, this is when the second meiosis division is complete, and now we have a true haploid female gamete. In the case of organisms like sea urchins and some cnidarians, it is in this state that the egg will actually be fertilized by the sperm. Thus, we can see that the eggs can be in different stages of development in the case of different organisms when the sperms can actually enter. To look at more detailed structures of the eggs, let's look at the mammalian eggs. Now, mammalian eggs, or the mammalian ova, are surrounded by a layer of cells that are called as the cumulus, and their role is to mainly nurture the egg. Now let us look at the figure at the right in the blue box. And when we look at this figure, close to the center, there is a circle. And I'm going to outline this circle in this blue circle that is there. That is the location of the ovum. All the small specks around it are actually the cells, which are the cumulus cells. The innermost cumulus cells that are right near the ova or the egg is called as the corona radiata. Now there is an extracellular envelope surrounding the egg that is rich in glycoproteins, and it is called as the zona pellucida. In the case of invertebrates, this region is also called as the vitelline envelope. And we can see that in the left side of the figure, there is a label that refers to the zona pellucida. Now, the gel-like cytoplasm that is right beneath the cell membrane of the egg is called as the cortex. The cortex is very rich in actin microfilaments and can form microvilli on the cell surface that can aid the entry of the sperm into the egg. Inside the cortex, we also see granules called as cortical granules that contain digestive enzymes. So the cortical granules are similar to the acrosome that are present in the sperm.
With this, we come to the end of our talk where we learned about different structural aspects of a sperm and an egg.